Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 20th July 2020. If you recollect from your memory, we were talking about a chapter called Human Health and Diseases. In our previous sessions, we discussed various diseases, causative organism, prevention, and possible treatment. And in our last session, we talked about a dreaded disease called AIDS, acquired immunodeficiency syndrome. And in this session, we are going to talk about another deadly disease called cancer, for which, as on today, there is no proper suitable treatment. Hence, we need to be careful. Prevention is better than cure. Remember, always being biology boys, prevention is better than cure. So let's go and discuss about this disease called cancer. What is cancer? We say an abnormal and uncontrollable division of cells is termed as cancer. I'll repeat, an abnormal, it is not normal, and uncontrollable, it is not under control. Uncontrollable cell division leads to cancer. And here you can see cancer, also known as malignant tumor, is a group of diseases involving abnormal cell growth with the potential to invade or spread to other parts of the body. So if one cell becomes cancerous, the cancerous cell spreads the cancer to neighboring cells, thereby tissue, organ, and entire organism. That's why it's very, very dangerous. It is considered as one of the major causes of deaths all over the world. Due to its severity, process of oncogenic transformation of cells, its treatment control requires most intense areas of research in biology and in general medicine. Cancer can be induced by external factors called carcinogens. Now, there are genes. What are carcinogens? These are genes called cellular oncogenes. So, carcinogens are the genes that cause cancer. So, carcinogens are responsible for cancer. They are called CONC, or proto-oncogenes, present in normal cells. These present in normal cells get activated under certain conditions, leading to oncogenic transformation of normal cells into cancerous cells. So how do you differentiate a normal cell with a cancer cell? Cancer cells, as I said, divide uncontrollably. Normal cells divide under controlled manner. Mitosis is under control. Second, cells do not show, what is important is contact inhibition. Cancer cells do not show contact inhibition, whereas normal cells, they show contact inhibition. Contact means be in touch. Inhibition, opposite to encouragement. Inhibit means discourage. In cancer cells, lifespan is indefinite. Normal cells lifespan is fixed, definite. These are the three major differences between a cancer cell and a normal cell. And most cancers worldwide, and people suffering with that. Women, of course, the breast cancer, India is leading unfortunately in that. Lung cancer, stomach cancer, cervical, again very, very high in numbers, and colorectal. In case of men, esophagus cancer, lung cancer, stomach cancer, liver cancer, prostate cancer, and colorectal cancer. The number of deaths, of course, way back in 2000, but those now we are in 2020, these numbers must have gone double, and they are very, very scary. That is the reason why I said we must be very careful with this number of deaths and the severity. What causes cancer? Now, normal cells transformed into cancerous neoplastic cells by physical, chemical, biological agents. And these agents are called carcinogens. Now, three areas are responsible: physical area, chemical area, biological area. So the physical agents are X-rays. Whenever a person gets unnecessary exposure to X-rays, prolonged exposure to X-rays, gamma rays, non-ionizing radiations like UV rays, so the person is likely to get cancer. That means all the X-ray technicians, persons working in what we call radiation labs are all under threat. And chemical agents, of course, smoking, all of us know, causes lung cancer. So tobacco smoking is very, very dangerous. There is a chemical substance called sodium azoate, very, very cancerous, carcinogenic. Methyl ethane, of course, you know that there was a leakage of this methyl ethane recently, and that is very dangerous. Methyl ethane, sulfonate. And biological agents, of course, there are cancer causing viruses. These are called oncogenic viruses, and these oncogenic viruses will have cancer causing genes inside their body. They are called viral oncogenes. These viral oncogenes by mistake enter into our body, person becomes cancerous. And then just now I said cellular oncogenes, C1s, are proto-oncogenes. In normal cells, when activated, how do they get activated? By continuous exposure to X-rays, gamma rays, UV rays, like that. Under special conditions, when activated, lead to oncogenic transformation of normal cells. Means normal cells become cancerous cells. And mechanism of transformation of cancerous cells, look at the diagram, photos, normal cells, and cell movement and cell proliferation. Look at the number of cells that are increasing, and this increase is uncontrollable, and the cells are becoming uncontrollable because of lack of contact inhibition. That's why normal cells show property called contact inhibition, means inhibits uncontrolled growth, whereas the cancer cells, they lost that property. Contact inhibition property is lost. As a result of that, cancer cells continue to divide uncontrollably, giving rise to mass of cells, and those mass of cells are called tumors. And tumors on subsequent stages becomes cancerous. This is how the proliferation takes place. Look at that, loss of contact inhibition and contact inhibition. See the difference? Cell to cell adhesion here, contact inhibition, and disrupted cell to cell adhesion, loss of contact inhibition. Look at the mass of cells that are being produced, and look at the difference in the structure when we see under electron microscope the normal cell structure and the cancerous cells. And I want you to see mainly on the cytoplasm of it. Look at the cytoplasm of it in normal cell and the cancerous cell, and look at the nucleus, the position of the nucleus, nucleolus. Okay, so how do you differentiate between a normal cell and cancerous cell? In normal cell, large cytoplasm, as you are seeing, single nucleus, yes, and single nucleolus, and fine chromatin, good chromatin. And cancerous cell cytoplasm has decreased, content has decreased, small cytoplasm, volume has come down, and multiple nuclei. The cell is becoming cenocytic, many nuclei, and multiple and large nucleoli. And then coarse chromatin has become coarse, and a tumor is forming among the normal cells. Look at that, the cancer cells dividing, and normal cells are looking normal, and the cancer cells, having lost that contact inhibition property, are forming tumors, and tumors further become cancerous. And types of tumors, there are benign tumors. These normally remain confined to their original location. They don't spread to other location. So thankfully, the benign one is restricted within the zone. And whereas in the case of malignant, it's mass of proliferating cells. They're called neoplastic cells, neoplastic or tumor cells. So benign tumor do not spread, and cause maybe insignificant damage, little damage. But the malignant tumors are very, very dangerous. So they proliferate, they spread across, the cells grow very rapidly, invade and damage the surrounding tissues, and these cells actively divide and grow and form a tumor, and tumor becomes cancerous. So cancerous cells escape from the site of origin and moves to distant place by blood. That is the danger. Since blood gets supplied to each and every part, along with the blood supply, cancer cells also will go to different parts of our body. That's how the cancer spreads. Wherever they get lodged, make the normal cells cancerous. This property is called metastasis. So keep remembering this word, metastasis. In this, two words one must remember. In various competitive exams, these two will be asked. One is the contact inhibition and second one is metastasis. And then detection. How to detect that a person has got cancer? One is biopsy and histopathological study. And in biopsy and histopathological studies, the tissue or blood or bone marrow will be checked. So tissue, blood, bone marrow tests for increased cell counts, leukemias. And biopsy of piece of suspected tissue cut into thin sections is stained means colored, and examined under microscope. This is histopathological study by a pathologist. And then we go to the radiologist. Radiography like X-rays, CT, computerized tomography, CT scan we say, used to detect cancers of the internal organs. Computed tomography, CT scan, uses X-rays to generate a 3D image of the internals of the object. And MRI, magnetic resonance imaging. Again, keep remembering this, MRI, magnetic resonance imaging, uses strong magnetic fields and non-ionizing radiations to accurately detect pathological and physiological changes in the living tissue. So through MRI we will come to know, like in physiology we see the concentration of the cytoplasm, we see the number of nuclei and number of nucleoli and the structure of chromosomes, chromatin reticulum. All this can be studied under MRI scan. And antibodies against cancer specific antigen. Antibodies against cancer specific antigens are used for detection of certain cancers like carcinogens, and the person is advised to prevent exposure. So we can count the number of antibodies, and when antibodies are becoming more, that's also an indication that the person is having cancer. So techniques like radiography, CT scan, MRI to detect cancers of the internal organs. Detection of cancer of specific antigens, and molecular biology techniques to detect genes in individuals with inherited susceptibility to certain cancers. So the common approach for treatment, once we come to know that there is a cancer in a person, how to treat, we have got only as on today 3 methods. One is surgery, removal of cancer cells using surgery. Of course there is a risk involved in that. Even if in the surgery, even if by mistake one cell is lost, metastasis will take place, and that cell will go to metastatic cell will go to another neighboring healthy cells and make them converted into cancerous cells. So surgery may not be that useful. Then radiotherapy, destruction of cancer cells using radiation. Of course there is side effects always. And then chemotherapy, destruction of cancer cells using drugs, anti-cancer agents. But again side effects, people say it is one of the worst treatments that can be given to a cancer patient, because more than the cancer, it is the damage done to the normal cells also will cause heavy damage to the person. But then we have no choice as on today except relying on biotechnology, and that is a long way for us still to go. So basically we have got surgical removal of tumor and irradiation of tissue, cancerous tissue, to kill the cancer cells. And of course we have got immunotherapy using interferon to boost cancer cell killing. That that is also nowadays interference are being used. As we said, look at that. This radiotherapy, tumor cells, irradiate lethally, taking proper care of normal tissues, and of course, the surgery removing the tumors. And then this is chemotherapy. Kill cancer cells specific for particular tumors. Majority of drugs have side effects like hair loss and anemia, as I said just now. And then immunotherapy, alpha interferons. This activates immune system to detect the cancer cells and destroy them. This is a very good one. And most cancers are treated by combination of surgery, radiotherapy, and chemotherapy. So you can see here, first, tumor cell death. Number one, uptake of tumor cells by antigen presenting cells. They are called APCs, antigen presenting cells. And now you got that CD8 and T cells, and tumor antigen is incorporated, and the APC stimulation immunotherapy takes place. As a result, the T cells stimulating immunotherapy, and immunotherapy results in strong T cell response is generated, killing remaining tumor cells. So we are activating one through radiotherapy and making other cells to get killed. But as I said, a lot of side effects. And how to avoid all this? Have a nice exercise, do a lot of exercise, sweat out your cells, and then have proper sleep. I said 7 to 8 hours of good sleep you should have in a day, in 24 hours. Eat healthy foods. Always you must have balanced diet and fresh foods you eat. Prefer to have fruits, milk, honey more than anything else. And then most important thing is stress-free life. Don't be under stress. De-stress yourself. Do yoga, meditation, pranayam, and so on and so forth. And there are various methods through which we can de-stress ourselves. So once we attain all these, I think cancer will not be attacking us. Our immune system will boost itself, and once our immune system is very good,